Hey, welcome everyone. Thanks for attending tonight. It's an evening here, but not so much for Dr. Lauren. We want to welcome everyone to the continuing series of new researchers and upcoming research that's going to be done on choroideremia and its carriers. And I just finished a lovely conversation with our presenter tonight. And I just was so excited because your accent is amazing. I think I could just you could record something, anything actually, and I'll just listen to your voice. It sounds awesome. She was a little bit, we were talking earlier today and she wasn't sure because she has a little bit of cold, but it still sounds amazing. So we're super excited to learn about your work, studying our perspectives, people with IRDs, when we have an opportunity to learn about gene therapy, what's our reaction to that, which is really interesting, as well as I think one of our first presenters, maybe our second, on female carriers. So I think there'll be some ladies in the house here that will be joining us tonight and learning about that perspective. So we're just really grateful. We have tonight Dr. Lauren Ayton, who's going to be here. She comes from Melbourne, Australia, and she'll be sharing her work and with her team. We want to thank our sponsor too, 4DMT, for hosting these. And once again, for those maybe that haven't attended, my name is Beth Voss. That's in my notes, but I didn't say that. And I'm the Director of Operations. So during Dr. Ayton's presentation tonight, she'll be conducting a slide presentation. And so if you have a question about any of her slides, feel free to use the question and answer or chat feature. I'll monitor that and I will gracefully interrupt, or we'll wait to the end and ask all the questions. And I think that's it. Share with us a little bit about what the weather's like down under. Tell us about what it's like living in Australia.
Well, thank you so much, Beth, for that kind introduction. And yeah, apologies to everyone in advance. I do have laryngitis, so maybe, I'm not sure if they can hear me. I'm hoping so. Yeah, so I have excellent. I have done every single possible, way of getting my voice back this morning. And I think honey and lemon tea has been the success. So it's an absolute pleasure to join you all today and to share some of the research that we're doing in Melbourne. To start off with, I actually thought I might show you some pictures of where we're based. Well, actually, before I get into the pretty pictures, I should just quickly give some disclosures. So I'm involved in a number of clinical trials in Australia, and these are the companies that I work with. And I'm also a consultant for Novartis and Apellis and have funding from a range of organisations, including the Choroideremia Research Foundation, which we'll talk about today, which we're very grateful for. Excuse me. So this is Melbourne, Australia. So whenever people think of Australia, they do automatically think of kangaroos and koalas, but Melbourne is quite a bustling city, and our research group is split between the Ioneer Hospital in the middle of Melbourne and also the University of Melbourne. And we're very lucky to work very closely with the ophthalmologists and other clinicians in this space as well. So if anyone ever comes to Melbourne, please call out to us. We'd love to welcome you and show you around. So in terms of inherited retinal diseases in Australia, we think that there are similar rates to the US and the UK. And this is quite an interesting question, actually, because we don't really have fantastic prevalence data. And that's something that our team are working on improving. But we think there's around about 16,500 Australians that live with an inherited retinal disease. And similar to other places, the most common of these is retinitis pigmentosa or rod cone dystrophy. And in terms of the macular diseases that affect your central vision first, one called Stargardt disease is the most common in Australia. And you can see there, if you look at the pie graph on the slide, that choroideremia is obviously present in Australia, but as everywhere else is quite a rare one of these conditions. And so we are running a program in Melbourne that's called VENTURE. And so this is basically a registry of people that have inherited retinal diseases of all forms, including choroideremia. And we are bringing in people basically to learn more about the clinical presentation of these conditions, as well as the genes that can cause these conditions and how they may affect prognosis and natural history. And we're also very interested in what we call patient-reported outcome measures. So this is really just about asking people that live with these conditions how they affect their life. And so we do a lot of work trying to compare the stage of a condition to what it actually has in terms of impact on everyday living. So for those of you who are able to see my slides, up on the slide, I have a picture of a back of an eye of somebody that has choroideremia. And what you'll see is a central dark pink area, and that's sort of the relatively healthy tissue. And then out towards the edges, you can see it looks kind of yellow. And that's because the retina out in that area has become atrophied, or the cells have died off. It means we can see through more easily to the white at the back of the eye. And that's why it looks brighter. And so we can take these very detailed pictures of the eyes and then compare them to the genes that we can detect in people from a blood test or a saliva test, and then also look at obviously daily impact as well. So I mentioned before that I'm an investigator on a number of clinical trials. So one of the points of Venture is that we can then find people for these trials. And I'm sure all of you on the call are probably aware of Luxturna, which was the first ever approved direct-to-patient gene therapy. And that's actually for any disease, not just for the eye. So there's definitely a space where ophthalmology is leading the field. And Luxturna was a very interesting lesson for us when it hit Australian shores, because we did have to very quickly try and find people that were candidates. And so this is one of the reasons that Venture exists now, is to try and make sure we have a database of people that are phenotyped, so we know what their eye looks like, and they're genotyped, so we know what gene they have. But most importantly, we know whether or not they want to be notified about these trials. So then we can let people know when something comes up that might be suitable for them. And then the other cornerstone of our program in Melbourne is working in the lab. And so the image that I have on this slide here is one of our collaborators, Professor Marnie Blewett, who's at the Walter and Eliza Hall Institute in Melbourne. And I'll talk a bit more about the work we do with them in a minute. But we do a whole range of preclinical studies, from developing new treatments to learning more about these conditions and how they actually progress, and also looking at very clever ways of looking at blood biomarkers to try and determine which people are at most risk of losing vision. And again, I'll talk a bit more about that later, because this is a very exciting part of our research, which has just been funded by the Choroideremia Research Foundation. So we're very excited to share with you what we're going to do in that space. So this is the core team for Venture. And I should say that we're very blessed. We have a really a large team across both this natural history registry and also our clinical trials unit, which runs a lot of the work. But in terms of the Venture registry, we have a research fellow, CC Britton-Jones, study manager, Sashini and Parker, who works as a data coordinator for us. And Tom and I are co-leads on this particular study. And I should mention Tom Edwards is a vitreoretinal surgeon who is an expert in gene therapy delivery. And so I've got some quite detailed slides up here again for those of you who can see it, and for those who can't, what the slides are showing really is the breakdown of what types of inherited retinal diseases we have in our registry. Now, I should say that this is a very biased cohort, because we are actively looking for people for trials that are coming up in the near future. So, for example, there is a condition called Bietti crystalline dystrophy, also known as BCD. And this is a very rare form of inherited retinal disease, but we're working on a treatment in Melbourne. And so we've obviously been actively searching for people with that condition, which is why there's a slightly higher number of people with that than would be normally. We have about 5% at the moment of our registry are people with choroideremia as well. And the other thing that's really important to know is that our registry includes everyone, so people of all levels of vision. And that's from people that have very early stages of vision loss and have really basically normal vision, all the way through to people who have no light perception left. And the reason that this is important is because there are different treatments and adaptive technologies coming up for people of all stages of vision loss. So it's very important to have them all involved in these registries. So I mentioned just then about treatments, and this is a fantastic figure by Cece. It's the fellow I just mentioned before. And basically what this image is showing is some of the treatments that are emerging. And the ones that I've highlighted at the start here are gene-specific therapies and DNA editing. So these are known generally as gene therapy. And you can see from that plot there, the dark blue-green colour is the people that it's most suitable for. And when you get to a lighter colour, it's less suitable. So basically for the gene-specific therapies, we think that we need to target people at an early stage of disease. So this is really, if we can, potentially before they start noticing symptoms or when they just have early symptoms. But then there's other treatments coming, and this includes things like RNA therapies and translational read-through therapies. And these are more for people kind of in that early to moderate stage of vision loss where, you know, there still is a significant number of retinal cells that are still working, but they're a little bit more advanced than perhaps for gene therapy. And then for people that have really lost a lot of vision, the challenge with things like gene therapy is that a lot of the cells have been lost. And so things like gene therapy don't work as well because you don't have the cells remaining to reactivate. And so that's when we start looking at things like stem cell-based therapies or the bionic eye, so prostheses. And there's also a technique called optogenetics. And this is a very clever technique where you're using a gene therapy kind of process to change the function of the cells in the retina to make them more responsive to light. And that's, again, very exciting novel therapy coming up. So I wanted to show you a bit of a quick video of what it actually looks like when somebody does a gene therapy treatment. And I'll talk you through the slide for those who can't see. But the thing to remember when you're watching this video is that the retina is incredibly fragile. So it's about the thickness and the fragility of a wet piece of tissue. And so it's very, very fragile. And so this particular surgery is quite impressive. So in this video, our surgeon, Tom Edwards, is actually looking at the front of the retina through a colour image. And then he also has some scans, which are shown on the right-hand side of the slide, which give a cross-sectional view of the retina. And what he does to do the gene therapy is actually inject into the space between the retina and the back of the eye and put that genetic material into the eye. And it's done through a virus. So it's a virus that doesn't infect humans, it doesn't make us sick, but it actually allows us to get that genetic information into the eye. And it's a really amazing technique. And I mentioned before that there is one regulatory approved gene therapy on the market that's called Luxturna. This is for a form of retinitis pigmentosa that's caused by a gene called RP65. So it's a very specific cause of IRD that can be treated with this gene therapy. But there's obviously a lot of other therapies that are following close behind. And this is a systematic review that our team did, looking at which genes are really being targeted in this next wave of treatments. And the largest peak on this graph here is showing that the gene RP65 has had the highest number of clinical trials and scientific papers written about it. And that makes sense. This is the gene therapy that has now been approved. And so obviously a lot of work has gone into it. But what's very exciting for this group of people is that choroideremia is very closely following behind. And so this is the third most common researched gene in terms of gene therapy for inherited retinal diseases. So the really interesting thing with this is as these treatments are being developed, we need to find people that would like to have the treatments. And so the first hurdle that we face is that historically people didn't have genetic testing. It used to be very expensive. It used to be very hard to access. And so this means that a lot of people, and I'm sure some in the audience today, have been told in the past, you know, you have this condition, there's nothing we can do, we'll see you in 5 years' time. And so now it's a bit of a change of pace. We're now sort of seeing these treatments emerging. And the story has now changed to, well, we actually need to know people's genes. We need to know who might be suitable for these treatments, and we need to know if they're interested. And that's really the cornerstones of our research team. They're the answers that we want to get. So this particular image here was from a recent systematic review by CC and the team, and it's looking at genetic diagnoses of IRD across the world. And so what CC's done is basically looked at papers that have used next generation sequencing. So this is a newer form of genetic testing, and reported on different cohorts across the world. And the reason I've put it up here is just really to show the disparity. So you will see that in the US, for example, there's a lot of publications, there's a lot of data getting shared about what types of genes are causing these conditions. But embarrassingly for us in Australia, we haven't really kept up. And probably, unfortunately, without surprise, places like Africa also are a little behind in terms of sharing that information. And that comes back to resourcing and other issues. So one of the take-home messages for me from today, I guess, is anyone that's listening that hasn't had genetic testing done, if they do have choroideremia, I would say this is definitely worthwhile doing now. And this is very much becoming more and more common. So there's a picture here which is showing a plot of how many, again, in the literature, so in the scientific literature, how often are people reporting on genetic results for people with inherited retinal diseases? And importantly, it's looking at the solve rate. So this is basically, if you have a genetic test, how likely is it that we're going to find the gene that's causing your condition? And this is one of the reasons that it didn't used to be so common, because it used to be that the solve rate was around about 25%. So we would go to great expense and time to do a genetic test and then not really likely find an answer. Thankfully now the solve rate is much higher. So we're now looking at around about between 60 and 70% of tests will come back with a positive answer. And the other thing to be aware of is that even if it comes back and it's an inconclusive test, the labs that we send your samples to actually then will update us over time. So if they come back at the moment and say it's inconclusive, if down the track they determine that it actually is a gene that could have caused the choroideremia, then they will come back and update with that over time as well. So that's something to be very aware of. Now, I've got here a very busy slide, and I've titled it The Importance of Clinical Assessments. And basically the idea with this is that we have amazing technologies at our fingertips now that let us image and measure the function of the retina. And so we can do very wide view images of the back of the eye now, and that's what's shown here in pictures A and B. And that allows us to look at the changes in the retina that are right at the outside of the visual field. And that's really important in choroideremia because obviously that's where the condition starts. Another really interesting test that we have is called autofluorescence. And the images here in E and F show what we can see with this particular imaging type. And what it does is it shows us areas where the cells have basically died. So anywhere that's black, we know that the retina is not healthy. And we can then correlate that with, again, people's reports of their vision. Are they having trouble navigating? Are they having trouble with mobility, for example? And we can look at what the retina structure and function is and compare that with daily life. The other interesting ones on this slide are probably the panels G and H. So for those of you who are able to see that slide, you'll see it's sort of got a multicolored picture. And basically this is a readout from a piece of equipment called a microperimeter. And the microperimeter allows us to measure the retinal sensitivity at very specific points. So I'm sure many of you have probably done this test. It is unfortunately quite boring. You sit at a machine and you press a button every time you see a light flash. And what that allows us to do is measure each individual little dot on that retina. We can see how sensitive it is to light in that location. And again, this is really important for us, not just to look at how it correlates with things at one static time point, but in terms of clinical trials. So if we give someone with choroideremia a treatment, we can then use this as a way of seeing whether or not it slows down the loss of those cells in the retina, or whether it actually may even improve things for people. The other image is down the bottom. INJ is the other one that I wanted to point out in a bit of detail. So this is a fantastic tool called an optical coherence tomographer, and it gives us a cross-sectional view through the retina. So if you think of the eye from the front, from the area that we can see clearly all the way through to the back of the retina, it's basically like cutting through the different layers. And so in those images, the black part at the top is inside the eyeball. Down the bottom of the image is the white sclera of the back of the eye. And so it allows us to measure individual cellular layers in the eye. And this is obviously an incredibly sensitive technique, which really allows us to see how things in that retina change over time. So again, I've just got a couple of images up here to show the power of imaging. And so this is a colour image of the back of the eye. And for experts in the field, we can see that this is choroideremia, but it can be a little challenging to see where the areas of unhealthy retina are. But this is the same person with one of those images I talked about before called autofluorescence, and black areas are areas where the cells have died. And so again, we can very easily see the areas that are potentially not going to respond as well to something like gene therapy. So our VENTURE Registry is very much around collecting that type of information. So we're trying to learn more about vision, about the images, about the genetics. But the other thing that we were very, very interested in is, what do people with inherited retinal disease feel about all of this? So there are a lot of emerging techniques coming up, and there's a lot of interest, obviously, including in the mainstream media. But how do people with inherited retinal disease feel about these treatments? And so we ran a study in Australia called the Attitudes to Gene Therapy for the Eye study. And we had a fantastic response. So we had almost 700 people respond to the survey, which is around about 5% of people that have the condition in the whole of the country. So we were incredibly thrilled with that. So this particular survey was developed by a professor called Heather Mack, and I have a picture of her on the slide here. And so she based this really on her clinical experiences and what people were questioning her about gene therapy and where she thought some of the barriers might be. And so that particular survey was then developed and improved through a multidisciplinary working group. And then we put this survey out to all Australians that had inherited retinal disease. So what we found very interestingly was that most people would take up gene therapy if they had inherited retinal disease. So it was about 92% of people said, yep, I would do it. If it was ready for me today, I would definitely do it, or probably take it up. But what was a little bit worrying for us was that only 28% of those people felt that they had a good knowledge of gene therapy. And this makes sense, right? So the technology is advancing really quickly, and it's a bit of a battle to keep up in terms of letting people know what is happening, and that knowledge transfer from the labs and from the clinical trial sites out to people who will benefit from the treatments. So my team is very passionate about making sure that we can develop educational materials, not only for people that might get the treatment themselves, but also for the doctors that look after them. So some other interesting things that came up from our survey was we looked at what people had done for previous treatments, and we found that most people hadn't tried any treatments for their inherited retinal disease. Of those that had tried things, vitamin A supplements, herbal remedies, and acupuncture were the most common. We were also very interested in what the barriers were for receiving gene therapy. So we asked people what would turn them off, basically, from getting these treatments. And again, most people said that they didn't actually see any real barriers. But those that did, the most common responses were the cost of the treatment. And again, this is not surprising at all. I'm sure most of you are aware that gene therapies are very expensive treatments. Thankfully, in most jurisdictions, the government will cover most of the costs, but they are expensive. So that's obviously a concern to people. And obviously people are concerned about side effects. And I'm happy to answer any questions people might have on that at the end. But in general, retinal gene therapy is a safe procedure, but there's always risks with these sorts of treatments, especially when we're doing an injection into the eye. And so that is something else for people to consider when they're looking at these treatments. The other thing I really wanted to highlight with this particular survey was just the absolutely outstanding community involvement that we had. So the way that we got our questionnaire out to people was through all of the different patient advocacy groups in Australia. And I've put the logos of some of these up on the slide as well. And what they were able to do is obviously reach out to the communities and share what we were trying to do. And we just had an absolutely outstanding response. So to give you an idea, we were kind of hoping for maybe 70 to 100 responses. So 700 was outstanding. And so I just wanted to really say thank you to these amazing organisations for their involvement. Now, for those of you who are listening, I'm assuming most of you aren't in Australia. If you would like to give your input into this, we would love to hear from you as well. So we are now going to expand this survey out of Australia and look at people's perspectives across the globe. And we're going to be launching this in the next few weeks. We are literally just finalising the last bits of paperwork. And I'm really thrilled that the Choroideremia Research Foundation will be able to circulate that around to you. So you probably will see this in a newsletter at some point to come. Thank you very much in advance for any input you may have into this particular issue. Now, the next, actually, I might pause there actually and just check if there's any particular questions on that, perspectives on gene therapy topic before I move on to female carriers.
You know what? We haven't had any questions yet. You're doing a great job, and nothing yet. But if anybody has any questions, please feel free to use that chat feature or the Q&A. I think you should just keep going. This is great.
Amazing, amazing. It's probably wise to keep going in case my voice gives out as well. So the other area that we are incredibly interested in with my team is female carriers of choroideremia and also X-linked RP, X-linked retinitis pigmentosa. So again, I think some of you on the call probably got lived experience with this. What has historically happened with female carriers of choroideremia is that they've been told, look, you're a carrier, but you only have one copy of the gene mutation, hence you should be fine. And a lot of people have just been sort of sent off into the community again and not really followed up very closely. Unfortunately, we know that's not always the case. And so I've again got some images up here from some of our collaborators at Oxford University, and showing that the presentation for female carriers of these conditions can be really varied. So a lot of carriers don't lose vision. They end up, their retina stays quite healthy, and the images on the left reflect that. So this is a relatively healthy looking retina. You can see in the functional images halfway down that slide that it looks green, basically. So it's sort of near normal responses to microperimetry. So we have good retinal sensitivity there, and the retina looks relatively healthy. The images over on the right, unfortunately, are a female carrier who did lose vision from the choroideremia gene mutation that she carried. And so up in the top images, those black splotches are areas where the cells have all died. And you can see that corresponds to a loss of retinal sensitivity. So this lady did lose vision, and she had an impact on her daily life. And so this is really interesting. Why is it that some female carriers are affected and others aren't? And before I go into, what we're going to do, I'll go back just quickly to the science of how X-linked inheritance works. So basically, females have 2 X chromosomes and males have an X chromosome and a Y chromosome. And so when a embryo is formed, there's a random allocation from the mother's genes and the father's genes as to what sex chromosomes that embryo gets. And you can see from this figure here, what it means is that if there is a carrier mother and an unaffected father, her sons will basically inherit one of her X chromosomes. And so it might be the healthy one, or it might be the one that has the gene mutation. So it's a bit of potluck, 50-50 chance as to whether her son will have choroideremia or not. The other situation is when there is a father that has choroideremia, but the mother doesn't carry a gene mutation. And in this case, because the father is going to give his Y chromosome to his sons, we know that the son won't inherit choroideremia because she doesn't have the gene mutation on either of her X chromosomes. So it does get a little confusing sometimes thinking about this, but this is really why genetic counselling is really important in families that have choroideremia, because we can actually counsel you on what the odds are of passing it down to the next generation. However, it gets more complicated than that. So as I mentioned before, historically we thought, okay, carriers don't really exhibit much of a phenotype, so they don't really show the signs of the condition. But we know that's not true. So these are some images from our PhD student centre showing the different classifications of women that are choroideremia carriers. And you can see really, you don't need to be an eye expert to see that if you look from images A through to D, basically you get more and more of those black splotches and areas showing areas where the retinal cells have died. And so we can categorise people into 4 main different groups, looking at how severe the condition is in them. And as you would expect, as the retinal changes get more severe, then obviously vision is more affected and daily life is more affected as well. So that obviously brings the question of why, so if all of these women have one healthy gene and one unhealthy gene, why are some of them having normal vision, no real impact on their daily life, and other women are really losing a lot more sight? And again, I've got quite a complex image here, but the take-home message for this is that, unfortunately for us, cell division is very random. And so when an embryo is forming in a mother, if there are 2 X chromosomes, so if it's a female child, then there are too many X chromosome genes in that female. So the body needs to switch off one of those X chromosomes. And so it's basically done randomly. So in some cells, the X chromosome from the father is deactivated. In some cells, it's the one from the mother. And in some cells, and so then basically each time a cell divides, you kind of get this randomness. And so this is called X-inactivation or Lyonization. And it's actually named after the woman that discovered it, whose name was Mary Lyon. And so the take-home message here is, as the embryo is developing, this random switching off of X chromosomes really relates to what ends up happening in the retina at the end. So if you have a healthy X chromosome and an unhealthy X chromosome for the choroideremia gene, and you switch off all of the healthy one, then you're more likely to have disease. Whereas if it's the other way around and one gene mutation is switched off, then you're much less likely to lose vision. So we are incredibly thrilled to have been funded by the Choroideremia Research Foundation to look at this in some more detail. And so the take-home message is that because females are very affected when they're a carrier, it is likely that they may be candidates for gene therapy down the track. However, we at the moment don't know which females will go on to lose vision and which ones won't. So if it's a female carrier who's not going to lose vision, then we wouldn't run the risk of treating them, wouldn't put them under a surgery if they're never going to lose vision anyway. And so it's really important that we start to learn more about this X chromosome inactivation and trying to work out if we can predict which women are going to be normal vision and which women may lose vision over time. And so this particular project that has been funded by the Choroideremia Research Foundation, we're going to look at the vision, and we're going to take images of the retina and do all of those scans I was talking about before, measure the retinal sensitivity, do the whole works, and then also take blood samples, saliva samples, and cheek swab samples from these women. And we're working with some incredibly talented people at an institute called the Walter and Eliza Hall Institute in Melbourne, and they are experts in looking at this X-inactivation. So they have these fantastic new techniques where they can actually analyse those samples that we provide them and get an idea of the skewing. So how many of the healthy chromosomes and how many of the unhealthy chromosomes are present within that sample. What our ultimate aim is that we might be able to develop a blood test or maybe even a saliva test, depending on what our results show, that could determine a woman's risk of losing vision later in life. So what that means is then potentially these women could be given a blood test when they're kids. So if it's a family where we know there's a history of choroideremia, we could give them a blood test at a very early stage, work out what their likelihood is of losing vision, and then decide whether or not to go ahead and treat. So we're really excited about this particular project. And again, just really wanted to thank the Choroideremia Research Foundation for the funding that's going to make this possible. As you can imagine, it's brand new, spanking new technology is very expensive to run. So it's a huge help to us. One of the other aspects of these female carriers is looking at what the long-term changes are for these women. And this is something that hasn't been done before. So there's been no studies that have looked at how the retina changes and how the vision changes for females that are carriers of choroideremia over time. So we have a fantastic PhD student, Senna, who I believe might even be on the line. And Senna is currently over in the UK at Oxford University and Anglia Ruskin University in Cambridge. And what she is doing is actually seeing some women that her co-supervisors, Dr. Tom Edwards and Associate Professor Jasleen Jolie, saw almost 10 years ago. And what we're doing is repeating tests that we did at that time to see how things have changed for these women over time. We can then again compare this to their genetics, we can compare it to what their vision looks like at baseline, and we can get some understanding of how things change for female carriers. And the real importance of this is making sure that we can counsel women more appropriately. So rather than saying, you're a carrier, don't worry about it, we can say, well, look, this is the evidence. We know that you show these sorts of signs. Therefore, we think that you are likely to have this sort of experience with choroideremia. So this is a very exciting piece of work, and actually was also supported very kindly from a Choroideremia Research Foundation travel grant, which allowed Senna to go across to the UK to do that work. So we're very excited to share the results of that with you once we've finished the analysis. The other piece to this puzzle is stepping aside from the clinical and the genetics and all of those sorts of things and actually talking to female carriers about their experiences. So this actually was something of interest to us, and the interest was actually then cemented from a Facebook post, which I believe was actually hosted by Choroideremia Research Foundation, where there was a discussion between female carriers about what their experiences were. So Senna, as part of her PhD study, has actually put together a survey which is looking at what the female experiences are if you're a carrier of choroideremia. So this includes things like what your perspectives are on gene therapy and treatment, what your symptoms are, so what sort of things you experience, but also things about how it feels to be a carrier of these conditions. And what we're hoping is that the information that we get from that survey can help to really direct better care for carriers down the track. And as a mum myself, I know how challenging this can be. You know, you're obviously often dealing with a male relative that may have choroideremia, and often your side of things gets pushed a little bit further down the line. But we're really, really keen to make sure that female carriers are part of this discussion and part of the potential audience and recipients for gene therapies and emerging technologies as we go forward. So this is an international survey, this one. So if you are interested, I've put a QR code up on the screen and also a URL link to the carrier survey. And we would love to hear from any female carriers. So whether you're on the call or whether you're a male on the call who has a female relative that might be interested, we would love to hear from you. The survey doesn't take too long itself, but it's really giving us incredibly valuable data about the female experience of choroideremia. So in terms of next steps for our research, we are doing a lot in the choroideremia space. So as I mentioned before, we are focusing quite a lot on genotype and phenotype. So we're trying to learn more about what the genotype means for prognosis and whether we can find different things in the retina that kind of help us give people a little bit more of an understanding about what their likely journey will be. As I mentioned, that the PhD that's being run by Senna is very much focused on female carriers. And part of that is building up a registry of female carriers that we will then continue to see longitudinally. So we're hoping to collect 10, 20, 30-year data on these women. And again, feeding back in not only to new trials and treatments, but also to standard clinical care. We're very interested in patient-reported outcomes. And so these are really surveys that we develop, which are specific for inherited retinal diseases. And the reason that they are so important is that the Food and Drug Administration, so the FDA in the US, and other regulatory bodies want to actually see that when we run a clinical trial for a new treatment, it's actually improving life for people. So it's really important that we develop questionnaires which make sense. And I'm sure, again, some of you in the audience have probably done questionnaires in the past, which have just been irrelevant to what your experiences are. So there's a lot of work going on to make these more IRD relevant. Of course, clinical trials and new treatments are something that we're passionate about and constantly working towards. Our site in Melbourne is actually part of the Foundation Fighting Blindness Global Consortium, and our aim really is to share information across the globe. And so we're really working very hard to make sure that everything that's done in the choroideremia research space is equal across different countries, and that we can kind of group our data to make it more powerful. And the last little aspect of our research that I've highlighted here is around new imaging biomarkers. And I wanted to end with a particular image just to show you, I guess, how exciting some of this technology is getting. So this image here is a technique that's called adaptive optics. And so adaptive optics is a way of taking photos of the back of the eye that get around all of the aberrations. So as you can imagine, when you're taking a photo through the eye, it gets quite distorted by all of the media and things in the way. So adaptive optics works around that, so you can get cellular resolution images. So at the bottom of the slide here, we've got some black and white images that show little black and white dots. And so these cells are basically in the retina, and we can image individual cells in the retina. So if you think about that, it's actually very mind-blowing. So it's a living person sitting there in the chair, and we're actually looking at their individual cells. But this is really important in choroideremia, because this paper that came out last year was looking at what the changes in cells are in people that either have choroideremia, and that's that top black and white bar there, or if they're carriers, or if they've got no choroideremia gene mutations in a healthy control. And you can see there that the dots down the bottom in that control situation are very small. And as you get an affected retina, the cells are a lot larger. And so this is really important information for us when we're developing new treatments, and also could be a biomarker for these new clinical trials as well. So that's sort of really where I wanted to leave it today. I'm hoping that was quite a useful overview for you of what we're doing here in Melbourne. I really wanted to end by highlighting our fantastic team. So we've got an image here of our team just at our last Christmas party. In particular, Tom Edwards, who is the tallest person in that image. is our VENTURE retinal surgeon who does a lot of these treatments. Fleur O'Hare in the front is our unit manager who runs our clinical trial space. CC, I mentioned before, is our postdoc fellow who's done a lot of this work, and she's in the red dress there. And Senna, who is standing behind the person with the antlers on. She is our PhD student doing a lot of work in this choroideremia space. We are incredibly blessed with a very strong team that are really working to learn more about these conditions. And I really am excited to share some of our data as we go through with the studies I've talked about today. I'm going to end with a little plug again for that carrier survey. So again, if you yourself fall into that category or you know someone who does, I've got a QR code there, and also very happy to send it to you after the talk. My email address is at the bottom, but we're really trying to gather as much data as we can from as many people as we can in this space, so we can design better treatments and implement them better into the health system. So thank you so much for all of your time this evening for you, and happy to take any questions you may have.
Hey, that was great. That was awesome. And we have just one question, so I'll give some time here. We'll start off. Renato is asking here, and then I have some other questions coming in. It says, hi, Doc.
Hello.
A little bit here. It says, hi, Doc. My name is Renato, 40 years old, and I'm the only one in the family who has CHM. What can you say about this and how I got? I'm in Seattle, Washington.
Yeah. Thank you so much, Renato. Look, I think it's an interesting issue. So we talk a lot about how choroideremia is X-linked, but there are definitely what we call de novo mutations. So sometimes the mutations just spring up and it's not just bad luck. So it can be a case where there may be a carrier in your family that wasn't detected. And part of that is that the genetic testing methodologies have got a lot better now. So sometimes people were tested a while ago and had a negative result, but actually may have been positive. Or unfortunately, it may have just been bad luck. And unfortunately, when you start thinking in detail about genetics and the number of letters we have in our genetic code, how easy it is for one of those to go wrong, these things do happen, unfortunately.
We have an anonymous attendee, a brave person, who's asking the question, is there a similar registry being created in the United States?
Absolutely. So I mentioned before the Foundation Fighting Blindness. So that is a US-based consortium, and there's sites all across the US. And what they are doing is so each individual site will likely have a registry as well. But the Foundation Fighting Blindness is trying to get all of these different sites to use similar protocols when we're measuring vision and taking photos and things like that, and then having a national registry. So I would definitely recommend having a look at the Foundation Fighting Blindness Consortium, and I'm very happy to send through some links as well. The people that are on that consortium, the different sites, are also doctors that are very interested in these inherited retinal diseases. So that's the other advantage with going to one of those sites. You know that you're going to be seeing someone that this is their passion. This is what they really want to work on.
I think that it's been kind of interesting, and I'll wait to hear if anyone else has thoughts on this. With our more recent conferences that we've had, we're seeing more people, more women that are attending that are affected. At least to my eyes, so what I see, it looks like there are more women that are affected. So this is incredibly interesting and something that we're very interested in. But the Lyonization process, I think that's correct, really did help explain well how that came to be. Is there anything that your trends that you're starting to see, or is it just more that we have the technology now to track it? Has there always been this many women affected, or is it increasing, do you think?
Yeah, it's a great question. So we think that it probably will have always been similar. It's just that I think, unfortunately, in the past, women may have been brushed off a little bit if they were having mild symptoms. Because the other thing to be aware of with these carriers is that they are more likely to have mild symptoms. So it is rarer for them to have the more severe kind of male type choroideremia. And so, I think there was a bit of that, probably women going, oh, well, it's, probably imagining it. But we're getting much better with our imaging. We're getting much better with our genetic testing. And so those sorts of things are obviously, again, raising awareness for us. The other thing that would have happened probably in the past before genetic testing got better is if there was a female carrier of choroideremia and she had really, significant vision impacts, before we did genetic testing, it probably would have been thought that she had a different form of IRD. Mm-hmm. So it probably would have been, thought that she had a form of retinitis pigmentosa, for example. Whereas now we can obviously do the genetic testing and say, no, it is the choroideremia gene. Therefore, this is the situation.
So many of us, I think I'm speaking for others that are attending today, we have, like myself as a carrier, we have fathers that have choroideremia, and then we have children, right? And we oftentimes focus so much on our sons who have it. We make sure that they're wearing sunglasses and they have their caps on, and are they taking all their supplements? And many of us, me included, I really haven't ever taken anything for my own vision. Do you think that we should follow the same protocols as we give our children, or would there be something that you're seeing that we should do maybe differently?
That's a great question. I think it's, again, one of those unanswered questions at the moment because we just don't know enough yet. And I think, and again, it's sort of this, we're very keen to raise awareness, but also not cause alarm because, again, most carriers won't have any significant impacts, and most carriers, if anything, might have mild changes. So it's unlikely that they're going to get sort of the more severe changes. But yeah, you're right. I think as we learn more and if we can identify women that are at higher risk, that's when we then start saying, right, well, let's put some of these, protective strategies in place just to really limit that sort of risk.
We have about 40 women, not all that attend every month, but of carrier women in a mom support group. So you would be more than welcome to come anytime. And and it meets at night, so it would work out well for you in the time.
Perfect. We'd love to do that. And maybe Dob in Sinner to come and talk about her work because she's a fantastic presenter. But I think we would love to share what we're doing. That'd be great.
Now, your friend Eric, my friend Eric, is asking a question. I know you'll be seeing him in a few weeks. Thanks for this comprehensive review of our CHM research environment. Many of us that have received a genetic test result were told that the results confirm the CHM diagnosis. Should we go back to find out what is the actual variant, nonsense, splice, site mutation, etc.? Why is it important, like mRNA? Thanks again for your time and effort in CHM research. Eric Hartman.
Thanks so much, Eric. So that's an excellent question. So all of these emerging treatments that are coming up, we think that some of the different variants will be more susceptible to different treatments. So it is of interest to us as scientists to know what the variants are. But at this point, I don't really think it's particularly clinically relevant for you. So again, the good thing is that, if you have had genetic testing and your doctor has a copy of that, or in particular, if you're on one of these registries around the world, then, we'll obviously have that information about the variant itself. But at the moment, there's really no clinical benefit to having that information, but likely down the track as the treatments progress, then it may be of interest.
Here's one that came in. If the potential testing works to determine if a carrier would lose vision later on, would they potentially receive the same type of gene therapy that the men would be getting?
Thought, yeah, that is definitely the thought. So the way that gene therapy works is it's basically trying to restore the function of the gene that's not working properly. The beautiful thing with the eye is it's an enclosed space, and the cells in the retina don't divide after you're born. So basically, if you can treat the retina and correct that gene mutation, the theory is that it should stop that disease from progressing any further. So the thought is that if we could identify women that were carriers for this condition that were likely to lose vision, then yes, we could potentially give them the same treatment.
Well, I think that's it, at least unless anybody has any more questions. You have about 1 minute. This has been great.
Oh, absolute pleasure. And as I mentioned, my email address is on there. We're very happy to answer any questions people might think of later as well. So always here if any additional thoughts.
And we'll make sure that we get that out. Of course, Kathy and the leadership team will be the ones that will coordinate those efforts, but we'll absolutely make sure we get that out. And you'll be seeing her at ARVO at the end of the month down in New Orleans.
Absolutely. Looking forward to it.
Well, we really appreciate it. Thank you so much for carving out a time with us today in your day.
Absolute pleasure. I'm very glad my voice lasted as well.
Wonderful. Well, thank you again. And we look forward to seeing you again. And we're really excited that actually the women are getting a look at too, because I think it's very important.
Absolutely. Completely agree. Fantastic. Have a lovely evening, everyone.
Thank you. Thanks everyone for joining us. And we look forward to seeing you next time.